Welcome to Notes from the Lab, an ongoing series of conversations on the past, present, and future of the BCG vaccine. My name's Denise Faustman, and I'm head of the immunobiology labs at Mass General Brigham here in Boston. In this podcast series, we're exploring topics related to the many on and off target effects of the BCG vaccine. And my guest today is Annelise Barron, the Keck Associate Professor of Bioengineering at Stanford University. Welcome, Annelise. Welcome
Thank
to our
you.
show.
Thank you, Denise.
Yeah, we're good friends, and it's a wonderful time to have Annelise today because she's doing this pioneering work on Alzheimer's disease and the role of a protein called LL37. She's a strong advocate on the role of infectious diseases in the cause of Alzheimer's disease. And recently, she's also had a focus on BCG as it relates to Alzheimer's disease as well. Welcome, Annelise. So I have about 6 or 7 questions, and I thought it's a great time to share your views with everybody who watches these podcasts. So the first one is, what took you to California and what took you to Stanford in particular?
So, previous to starting at Stanford University in the Department of Bioengineering, in 2007, I had been a professor at Northwestern University for 10 years, and I was in chemical and biological engineering, and I was getting interested in more medical types of applications of the materials and um, strategies that I was developing. And I also, although I'm originally from Milwaukee, I've always, I'd grown up mostly in Seattle. I just felt like I wanted to be on the West Coast. So having done really well at Northwestern, getting my lab started, and I had risen actually to full professor at Northwestern by 2006. And then the bioengineering department at Stanford was just starting, and it was very interesting to me that it crossed the School of Medicine and
Mm-hmm.
Engineering. So I applied there. I was very lucky because there were 600 applicants, and two of us got positions. And it's really been an amazing place to be because I was immediately connected with clinical researchers
Mm-hmm.
in neuroscience and infectious disease, and I'm able to, interact closely with people in the medical school who are just a 5-minute walk away from my
Mm-hmm.
lab.
Which is amazing.
Yeah, it's a very
Yeah.
special kind of physical proximity that Northwestern did not offer. Their medical school is downtown I was on the undergrad campus in Evanston. So Northwestern is also nice, but I should say that Stanford is so good. It's such
Okay.
a great place to work and so supportive. So, that turned out to be a good move. Although they say, Denise, and it's very much true, you know, moving a laboratory is like moving a massive sheet of plate glass. You know,
Yeah.
it will break, and it really does take a long time to get things going again.
Yeah. Because it's
But
people, it's experiments, it's tissue culture
yeah.
goods, it's everything. It's everything. Yeah, it's worse than moving a house for sure. Okay, so tell everybody how you got interested in Alzheimer's disease.
Okay,
What
so
was that link?
I had started working on the host defense peptides, also called antimicrobial peptides, while I was at Northwestern. One thing my lab does is we develop mimics of helical amphipathic peptides. So these are, essentially designed to look biophysically very similar in their structure, their chemical and physical structure. So their charge, their helicity, their patterning of hydrophobic and polar, and antimicrobial peptides were a natural thing to look at because they are very powerful broad spectrum antibiotics. And in particular, there's a human antimicrobial peptide called LL37. It's 37 amino acids. It was discovered in Stockholm in the late 80s, early 90s, and as initially an antibacterial peptide, but it turns out to be antibacterial, antifungal, antiviral, and anti-cancer and antiparasitic and pro-autophagy. It's a very old peptide, like 300 million years it goes back
Mhm.
to, long before humans existed, as a class of peptides. But, it's kind of like the Swiss Army knife of your innate immune system. It participates
Mhm.
in many different signaling, immunomodulation, and biophysical functions. So I was interested in that peptide. I was mimicking it with this class of compounds called peptoids. So because I was an expert in antimicrobial peptides, I was on an NIH panel
Mm.
reviewing proposals. And I didn't know anything about Alzheimer's that disease yet. I just, at this point, I was just interested in infectious disease. But it just so happened that, this is a really amazing kind of light bulb moment or coincidence that I was, finishing up my last reviews before the NIH study section meeting the next morning in Washington, D.C. And it was like 2:30 in the morning. And
Of
I'm
course
just
it was.
I got an email from someone in my lab right at that moment, and we had just received some LL37 from a collaborator in the Netherlands, and my student had put the one-letter code 37 amino acid sequence in the email and said, "Oh, we're excited to start working with this as a comparator to our compounds." And so I sat and studied the sequence and noted some things about it. And then I turned back to the proposal I was reviewing, and it was about Alzheimer's and A beta. And it just so happened, like exactly that moment that the LO37 sequence arrived, I'm looking at the A beta sequence on this paper in front of me. And, I could look at the one letter code and I'm like, oh my God, these are complementary peptides. These will
Mhm.
bind to each other. I could see it, just because I can look at a one letter code and I can project the structure of it
How
in my
much
mind.
overlap was there? How many amino acid overlap?
There were some key motifs. It was more sort of general biophysical that LL37 is highly positively charged, like +6, and A beta is quite negatively charged, -3. But what was unique is that they both have this particular motif, and even though they're pretty short peptides, only about 40 amino acids, they both have this double phenylalanine motif,
Mm.
Phi. And
Hmm.
those are very rare, and they really like to interact. Phenylalanines like to interact with phenylalanines, and I could just see that those were going to click in, and indeed it has turned out that they do. So I started working on that. That was February 2010. So that's now 16 years ago. And so I just said, wow, that's so interesting if this what I thought of as an antimicrobial peptide and what I thought of at that time, A beta, as just an Alzheimer's amyloid peptide.
Hmm. Hmm.
Also, that's very interesting if they would bind to each other because they're both endogenous to the human, system. And then later, it has unfolded that A beta itself is an antimicrobial peptide too,
Hmm.
of a
Hmm.
sort, of a different class. And whereas and so we did go ahead and with collaborators in Italy, we studied the binding of these peptides, continued to study it. It turns out that the complex between them is a very potent, powerful antiviral. Um, but it also turns out that LL37 prevents A beta from forming oligomers and plaques, which
Hmm.
is kind of this hallmark of Alzheimer's. And so the way it has all unfolded is that we initially published that result in 2017. We published the first paper showing the binding by many, many different biophysical methods, and then continued to study it. And then it came out that same year, 2017. The strong connection between infection and Alzheimer's
Hmm.
started to
Is that when
really.
that literature started? That's what I was going to say. Did you sit down and do a literature search immediately and go, do
Yeah.
infectious diseases cause Alzheimer's?
Yeah, so what's really fascinating is there was a small group of renegade researchers that had been studying the connection between infection and Alzheimer's for like 30 years. So the real OG is Ruth Itzhaki in Manchester, England, and but she was ostracized, remained unfunded most of her career because she would not give up on this idea that the herpes viruses are involved in Alzheimer's
Hmm.
disease, which she was absolutely right, and she's vindicated. But it became so I got invited. I'm not exactly sure, but this researcher in Sherbrooke, Tomasz Fulop, somehow heard of me. He was also looking at A beta as an antiviral peptide at that time, a wonderful old physician who treats geriatric patients.
Hmm.
And he invited me to speak in a special symposium in San Francisco about infection and Alzheimer's. And this was the IAGG meeting, the International Association of Geriatrics and Geriatricians or something.
Hmm.
And
Hmm.
he had been trying to have a symposium on that topic for like 15 years. And it was the first year that he had proposed a special, 1 day symposium on infections and Alzheimer's and was allowed to have it. And so I spoke, and then the whole group of us that spoke, we got together and did a joint review right after,
Hmm.
published that, and included the, LL37 hypothesis. So at that point, though, I was really kind of focused on the idea that, it made an involvement of LL37 in Alzheimer's made sense because it was here we were saying that infection could cause Alzheimer's and it's anti-infective. Oh, and by the way, it binds to A beta and neutralizes A beta's
Hmm. Hmm.
effects. So we started there, but it's kind of like the experience has been extremely exciting and gratifying as a scientist because it's like putting a puzzle together, and every time we find another piece, it fits perfectly and the picture becomes more clear. Now, then I became interested in ways to upregulate LL37 in the human body because you can't really inject it because it's
Hmm.
like $220 a milligram.
Yeah.
And
And peptides don't last very
yeah,
long.
and
Right? Yeah.
peptides
Yeah.
degrade
Yeah. Yeah.
instantly, and your body automatically makes it. And then I started teaching a class in biotechnology, and I invited Christine Stabell-Ben to give a lecture.
Hmm.
And this was probably 2019 or so.
Hmm.
And I learned about BCG and how it has these astonishing beneficial off-target effects, which include protecting against infection. And
Hmm.
sure enough, I searched, and there are, a number of papers from disparate labs around the world showing that the BCG vaccine upregulates the expression of LL37 in the gut epithelium.
Hmm.
So this was shown with human babies that received BCG and OPV on birth, and then at 6 months they looked and saw really nicely elevated LL37 expression in those vaccinated infants. And
Hmm.
obviously, BCG is so safe because it's given on the first day of life in many countries, and I think it should be given here in the United States too. So then I applied for the NIH Pioneer Award based on that idea in 2019. And the Pioneer Award, for those who might not know what it is, it's part of a program at the NIH that is administered from within the Office of the Director. It's called their High Risk, High Reward program.
Hmm.
And it's an interesting proposal because you don't give aims. It's not like an NIH proposal
Hmm.
where you lay out the whole project with aims, method, da da da. But it's actually a 5-page essay.
Hmm.
And they're trying to find people that are kind of outside the box thinkers, willing to take risks, and they're trying to fund projects that may fail, but if they didn't fail, could really make a difference.
Yeah. Yeah.
So I proposed, I basically put in my essay, I said, Alzheimer's is caused by polymicrobial infection.
Hmm.
So, but a vaccine against any one of those microbes is not going to be the ticket, like a targeted vaccine,
Hmm.
because there are many,
Hmm.
and I'll talk about those in a bit. But, there is a vaccine which will just strengthen the whole immune system across the board,
Hmm.
help the body fight infectious disease. It does that by upregulating LO37 among many other
Yeah.
mechanisms. So it has many mechanisms of immune modulation, but upregulating LO37 is one. And um, and that is very meaningful for Alzheimer's linked to infection because LO37 also blocks A beta fibril and plaque formation.
Hmm.
So I applied and I I got it. Actually, an interesting kind of instructive thing here is that the reviewers totally didn't get it. I made it to the interview stage, so they get several thousand applications for like 7 awards when they narrow it down to like 21 that they
Hmm.
interview. So I did my interview, and then you get a really short, like one paragraph back from the reviewers that interviewed you. And it's very short. It's like 15-minute interview with 5 minutes
Yeah.
for questions, something like that. And this is for $3.5 million, right? If you include the overhead, it's a $7 million grant. And they wrote this thing like, what is this about a vaccine? Like they clearly didn't get it. Like they totally didn't get what I was trying to. But
Say,
interesting thing, I wrote to the program administrator and scientific review administrator. I wrote kind of a fiery response to the review because it was just so off base, and explained like the four
what you were
things they
trying. Yeah.
fundamentally gotten wrong, which they could only have gotten them wrong if they hadn't read my proposal, right? So, and you know what? They funded it. And then they established a new protocol at the NIH, which is you get to respond to your reviews.
Oh, that's a good idea. That's
Yeah.
a good idea.
And before, just for this high risk, high reward program,
Yeah,
right? So
yeah,
that's
yeah.
the pioneer, the transformative R1. And so now they let you have a 2-page rebuttal, which is awesome. And they look at that before they decide who they're going to fund. So,
So that's
that
so what do you think the state of the field is right now? If you went out to the Alzheimer's community, and I think it may vary per country, but let's talk about the US. If you went out there and said, I think infectious diseases could accelerate Alzheimer's or even cause them, what do you think the buy-in right now is? Do you think it's 90%, 50%, 30%?
I would say there are some people that really don't want to admit that infection plays a role in Alzheimer's because of their, investments in their careers and their and their
Mm-hmm.
consult jobs and all that. But at this point, everybody does acknowledge that the approach that's been taken in the Alzheimer's field, and in Parkinson's too, over the last 20 years, which is just drugs, the drugs that were previously just kind
Mm-hmm.
of ameliorate symptoms slightly. But now, then once they started targeting amyloid clearance,
Yeah.
A beta clearance, and now they're looking
Mm-hmm.
at tau clearance. They put everything they had into it. They had 460 plus clinical trials,
Yeah.
hundreds of billions of dollars. That basically failed, and everybody agrees that's failed. And then I would say the awareness that infection is more likely the root cause is probably, it's close to 100% that people are aware that this is
Mm-hmm.
moving ahead. And in terms of acceptance, I'd probably put it at 35% at this point. It's just like it's like being a sunshine patriot, or I'm the winter soldier. Yeah, most people don't want to they don't want to be seen as agreeing to anything that out is an outlier of the
Well, I
conventional
mean, that's
wisdom.
kind of common. The discovery process is brutal. But I have to say, I think in different countries it's very different. I have in the lab right now a Japanese postdoc, and she's a dentist and a PhD. And she says in Japan, everybody accepts that if you don't have good dental health, then
It'll,
you can get
yeah. And in
Alzheimer's.
fact, I should mention, tell her for me,
Yeah.
some of the very best work relating um, pathogenic anaerobic bacteria in the mouth to
Mm-hmm.
dementia, and just to, systemic dysregulation of the immune system and gut dysbiosis.
Mm-hmm.
Like beautiful work has been done in Japan there with animal models of
Yeah.
Porphyromonas gingivalis infection. And so I would say, we're right. And in terms of the BCG field as well, like, the truth is for the infectious etiology of Alzheimer's
Mm-hmm.
disease, for BCG, everything people need to know is kind of already out there.
Yeah.
And
Well,
so
it's hearing it enough to realize you got to put it on your list as it's a possibility.
exactly. What we have done at Stanford is we have created a model of Alzheimer's um, initiation, Alzheimer's like disease in mice by infecting their molar teeth with
Wow.
this
Okay.
oral. It's an oral, what you would call a pathobiont. So it's a bacterium that can cause pathology
Mm-hmm.
with the right circumstances, called Porphyromonas gingivalis. And
Mm-hmm.
gingivitis is named after that bacteria. And it turns out that this is really, it's like the coolest like as a scientist, I find this disease mechanism fascinating and fun. And
Hmm.
because you always say, well, why did we not like really uncover the root cause of Alzheimer's all
Hmm.
this time when it's been studied since, 1905, 1906, right?
Hmm.
That's
Hmm.
always astonishing because there are so many smart people all around the world who've worked on it. And I think the answer there's pretty astonishing complexity, and work had to be done along many lines by different types of experts to bring the story together. And I've just published this in a review article in the Journal of Internal Medicine at the end of
Hmm.
December. But essentially, the Porphyromonas gingivalis, that organism it's it's anaerobic, so it can't grow in the presence of oxygen, which is why it digs itself under your gums. So
Mm.
it makes
It's less and less
enzymes.
oxygen.
Yeah, it makes enzymes that degrade your tissue, the tissue that attaches your gums to the teeth,
Hmm.
and it tries to go deeper and deeper to get away from oxygen.
Hmm.
And it uses these enzymes called gingipanes. It also has collagenases that are proteases, that attack
Hmm.
this tissue.
Hmm.
And that bacterium, interestingly, only eats meat, basically. It eats amino acids and peptides. So it's a meat-eating
Oh.
bacteria, but it relies on Candida albicans to be virulent and to flourish because Candida albicans is an extremely voracious consumer of oxygen. So
Mm.
what happens is that
Oh, it
PG,
makes a better environment.
yeah, so P.
So, but you
gingivalis.
haven't answered the question, did the mice get mental disease?
Oh yeah, absolutely. You get
Oh,
all the hallmarks of Alzheimer's
that's
with a
amazing.
PG infection. It is, and that's been shown now by several labs, but with different ways of infecting the mouse. So
Hmm.
sometimes they paint it on the gums,
Hmm.
sometimes they gavage it into the mouth and down the gut, and they create infections in different ways. Yeah, and you get all the Alzheimer's pathology in wild type mice. You don't,
Hmm.
whereas there are Alzheimer's mice, but they're an extremely contrived model.
Well, I mean, that's been a problem in the field of
Yeah,
Alzheimer's, right? Like, do you trust the mouse models? They're not
you really
spontaneous.
can't because it's very over engineered. That being said, you kind of have to do it anyway because it's
Mhm.
what there
It's
is.
a
But
checkbox.
yeah, it's a checkbox, of course, a very expensive checkbox and always
Yeah.
in scientific research. Okay, so one way to prevent Alzheimer's until you can
Hmm.
get BCG is to cut all the carbs out of your diet and just eat carnivore. And it'll work better if you include sauerkraut. So that's my
Oh, interesting.
dietary.
Because
Yeah.
as we wind up this interview, I was going to remind you that there's a lot of lay people who listen to these podcasts.
Yeah. Well,
So
and so
everybody wants a take-home message of
yeah,
what
the
can I
take
do
home is
today?
you can prevent Alzheimer's if you're currently, cognitively healthy, you
Hmm.
can prevent Alzheimer's just by cutting all the carbs out of your diet. And those supplements I listed off that upregulate
Hmm.
L37 are also beneficial. Let me just finish quickly the disease mechanism. So, so here you have Candida and PG in the
Hmm.
mouth. All right. Now, when PG is in its virulent sort of state,
Hmm.
it these gingipane enzymes it releases, they completely turn off your antiviral immunity. It's gone.
Okay.
Okay,
Okay.
so and most humans are colonized at least by HSV1,
Hmm.
varicella zoster virus, human cytomegalovirus, Epstein-Barr virus. Okay, so
Almost
those four,
everybody by the time you're 30,
yeah, by the
20,
time you're 30, you will
all
have all
those.
of those.
Yeah.
Okay, so those viruses, herpes viruses, they're neurotropic, so they actually live in the neurons.
Hmm.
Okay,
Hmm.
so and this is where we're going to get to why BCG is so powerful. There are several mechanisms, several of them, but we just kind of uncovered a new one. So, there is no blood-brain barrier inside your neurons, and these viruses are inside your neurons. That's where they
Yeah.
live.
Some of them live long term. Yeah.
Right.
In neurons.
So Candida plus PG end up producing gingipanes, which you're also swallowing with your saliva. Then your antiviral immunity has been turned off. Now those herpes viruses, we all have some of them, if not all. They replicate inside the neurons, they reach the brain. Now, as they replicate and get into the brain, they cause inflammation. And in particular, they activate what's called the NLRP3 inflammasome. The
Hmm.
NLRP3 inflammasome has been very meticulously tied to
Hmm.
Alzheimer's initiation and staging. And so it just so happens that BCG, oh, and then I should mention A beta fibrils and
Hmm.
oligomers
Hmm.
also upregulate the inflammasome. And the inflammasome is what leads to neuron death. You
I
get the
see. I
expression
see.
of caspase 1, ASC, IL-1 beta, but most importantly, interleukin 18. And then that IL-18 turns on a cell death pathway called pyroptosis, and
Hmm.
neurons die. BCG inhibits that. BCG number one, it produces LL37, which keeps A beta in the monomeric state.
Hmm.
So now that doesn't cause inflammasome activation. And number two, um, BCG itself, it inhibits the inflammasome by other mechanisms as
Hmm.
well. So essentially, what I think you're observing, Denise, in your clinical trial at Harvard, is that you're seeing it looks as if BCG, given quarterly for 2 years,
Hmm.
seems to just halt cognitive decline if it was occurring
Hmm.
before. And
Hmm.
I want to say it makes perfect mechanistic sense that you would halt it and then you would start to allow healing. And you should even, in some people, if they're young enough and the damage has not gone far enough, they
Hmm.
should improve. We already have the prevention for Alzheimer's and Parkinson's in our hands, but it is really essential to deploy it. And that's going to take an investment and some years, but the sooner we start, the sooner it's
Yeah.
operating.
That's a nice way to tie up all these thoughts because prevention is one thing, treatment's something else. And as you know, we have some very provocative data that prevention in BCG may be very important for Alzheimer's disease. But let's not wait too long because we don't yet have the data that if you're end stage Alzheimer's disease, we can clear up or regenerate a brain. And it's kind of like cardiovascular disease. If you take Lipitor, it helps you from ever getting it. But Lipitor in the hospital the day you're having your heart attack is a little late.
The one thing I would say is, this is something we can do, and luckily there's a very um, supportive global community
Mm-hmm.
that studies BCG and continually are finding its benefits. And I just want to mention too something, researchers have even shown that it's not only is it safe for premature infants
Mm-hmm.
to get BCG
Mm-hmm.
on the first day of life, but it actually enhances their health and their survival.
Oh, absolutely.
So
I
it
mean,
I can't imagine something safer than something you could give to a preemie and make
yeah.
them better.
It's amazing because when you have something that has a 100-year history of safety, it's like nobody has those safety profiles. So it should be given more credit than something that's new that has only been tested in hundreds, not
So
millions.
here's the thing, like, they did some studies when we were having COVID. Does BCG,
Yeah.
prevent COVID? And, the devil, of course, is always in the details. Not every
Oh,
BCG, not every way of delivering BCG will prevent COVID.
and the
But
time after vaccine.
yeah, but your strains, so I think either Tokyo or Connaught strains
Yeah,
that you
very
have,
potent,
and
extremely
the
potent.
repeated administration.
Yeah.
Can
And
I ask you
also,
again?
it's different being a neonate than an adult, and the time course of drugs, we all know, is totally different. So, a benefit like TB, you give it as an infant, but you're looking for protection at age 12. You've had
Yes,
12 years for it to mold the immune system.
yes.
So, BCG is not a fast antibiotic for any of these
No.
diseases, but talk about good data for ultimate prevention. It's beautiful.
I just want to say to Denise, I want to thank you for the work that you have done throughout your career. When did you start working on BCG?
Over 25 years ago.
So I just want to, for the whole world to know, if Denise had not worked so hard with many obstacles in her way and much discouragement by people, people love new drugs that can be sold for a lot of money, and
Hmm.
BCG
Hmm.
is not one of those, right? It's
Yeah,
it's kind
yeah.
of open access in the vaccine world. You would think, although they seem to have made it impossible to get in the United States, which
Well,
is
you
very
can't get
strange.
it even globally. I mean, bladder cancer. But on that note, I have to thank you because this is our first lecture in this Notes from the Lab on Alzheimer's. And probably most people aren't aware BCG is being tested for Alzheimer's around the world, in Israel, US, both directly and indirectly in epidemiology studies. And Annelise is one of the leaders at trying to figure out why it benefits Alzheimer's. So if you want the nitty gritty on how mechanistically it could help a lot of people in cost affordable ways, reach out to Annelise. And we thank Annelise today for joining us, and your scientific contributions to this field are really innovative and fun, and certainly very practical for people. So, to our listeners, if you want more information on the overall podcast series or more on the BCG clinical trials, just go to www.faustmanlab.org or email Annelise directly. Thank you for your time today.
Thank you, Denise.